Microbial Metabolism, Part 1. Every cell wants to maintain its metabolic properties and ultimately divide. In order to do that, a cell requires nutrients which provide the cell with building blocks and energy. Cells face the challenge of transporting nutrients across the selective cell membrane and getting the proper nutrients into the cell. Once inside the cell, energy and enzymes are required to metabolize the nutrients into smaller building blocks that the cell can then use to make new cellular components. We will discuss each step in this process and refer back to this slide along the way. By the end of this lecture, you will understand cellular respiration and its importance to the cell. You'll learn the significance of reduction and oxidation reactions to metabolism, and then learn the stages of cellular respiration. Later, we'll briefly touch on fermentation and anaerobic respiration, cellular processes some organisms use when there is no oxygen available, and then end with a nice overview of metabolic pathways. Nutrients are transported into the cell and then broken down in a stepwise manner during catabolic reactions to produce cellular energy in the form of ATP. Aerobic cellular respiration requires oxygen to keep these metabolic processes going. Cellular respiration also provides intermediate products that can be used for synthesis or anabolic reactions. In multicellular organisms with lungs, cellular respiration is not the same as breathing, but there is a link between inhaling oxygen and cellular respiration. Cellular respiration occurs in different areas of the cell in prokaryotes and eukaryotes. Glycolysis occurs in the cytoplasm in prokaryotes and in the mitochondria matrix in eukaryotes. The Krebs cycle, also known as the citric acid cycle, is associated with the cell membrane in prokaryotes and, again, in the mitochondria of eukaryotic cells. The last stage of electron transport, also called oxidative phosphorylation, occurs across the cell membrane in prokaryotes, again, in the membranes of the mitochondria in eukaryotes. In the mitochondria, there are specific locations within that organelle where these processes occur. Energy can be classified as potential or stored energy, or kinetic energy, the energy of motion. Chemical energy is stored in chemical bonds made between atoms and molecules. When these bonds are broken, the energy that is released can be used or converted to mechanical energy that is used to do work. Organisms may be classified by the energy source they use, and also by the source of carbon used for their metabolism. For example, phototrophs get their energy for electron transfer from light, but chemotrophs get energy from breaking chemical bonds. Carbon sources are either found in organic or inorganic compounds. Organisms like plants and cyanobacteria are autotrophs, that is, because they convert inorganic carbon dioxide into organic compounds like glucose that can be used by the organism itself or other organisms that consume them. Auto means self, so autotrophs are organisms that are self-sufficient. They produce their own food. Heterotrophs are organisms like us that we get our carbon source from other organisms. We feed on autotrophs and we feed on other animals. So heterotroph means other. This is a good summary of the different categories and examples of the types of organisms in each. The energy sources and carbon sources of different types of organisms can be combined to give a better explanation of the energy source and carbon source. Also included here in this table are examples of the types of organisms that are in each category. There are two laws of thermodynamics. The first law states that energy cannot be created or destroyed, only transformed or transferred from one object to another. It often results in loss of usable energy. In this figure, light energy from the sun is transferred to the plant, and some energy is lost in the form of heat. The plant converts the solar energy into stored chemical energy in the glucose it produces during photosynthesis, which also results in a loss of energy in the form of heat. The stored chemical energy can then be converted to mechanical energy when the bonds in the glucose molecules are broken during cellular respiration in the animal, again releasing heat in the process. The animal uses the mechanical energy made during cellular respiration to walk and hunt, and again, heat is released during this process. The animal converts chemical energy to mechanical energy of motion. The second law of thermodynamics states that energy transfer will always result in a greater amount of disorder in the universe and a loss of usable energy. What does this mean? This simply means that molecules naturally move from a state of order to a state of disorder. Entropy is the amount of disorder of molecules, so it's a measurement of that disorder. In this example, the structure of the water molecules in the ice is very ordered in a lattice structure. The amount of entropy or disorder is very low, but as the ice melts, the amount of entropy starts to increase when the molecules begin to move around more and farther apart, and the lattice structure is dissociated. When the water molecules reach the highest state of entropy, the water molecules are farthest apart from each other and highly disordered. So progression of entropy from lowest entropy to the highest state of entropy does not require energy. It's what occurs naturally. But to go from a high state of entropy, very high disorder, to a lower state of entropy, low disorder, and more order, it takes an input of energy. For example, this might not be a great example, but it's one I use because everyone can relate. When you were a kid, it didn't take much for us to spread our toys all over the house. But it seemed like it took lots of energy to put those toys back in the toy box in a very ordered arrangement. So we can think of those toys as when they're all over the place, having a high state of disorder, very much disorder, very high entropy. But to put them back in the toy box, to collect them and make them ordered again, it takes a lot of energy. Okay, keep that in mind. In living things, mechanical energy, the energy to do work, is used to transport substances like nutrients across the cell membrane. I spoke of this in the membrane transport lecture. Mechanical energy is used to do mechanical work, like to contract muscles to move our bones, or in moving flagella to allow cells to swim. In this lecture, though, we're concerned with biochemical work, which is the synthesis of macromolecules during metabolic reactions. When molecules are joined by bonds to put them into a more ordered state, energy is required. Survival of organisms that undergo aerobic cellular respiration rely on organisms that are able to undergo photosynthesis and vice versa. In this figure, the plant uses energy harvested from the sun to convert carbon dioxide and water to glucose and oxygen during photosynthesis. The rabbit then uses the glucose and the oxygen to perform cellular respiration and produce energy in the form of ATP. Heat is released during this process. The products of cellular respiration, carbon dioxide and water, are then cycled back to the plants, and the cycle continues. During cellular respiration, glucose is oxidized to carbon dioxide and oxygen is reduced to water. But what does that mean? Oxidized, reduced. Oxidation reduction or redox reactions are seen throughout metabolic processes. These reactions involve the transfer of electrons from one molecule to another. Keep in mind, electrons have a negative charge. So the molecule that gains electrons during transfer is reduced because the electrons are making it more negative. The molecule that loses the electrons is considered oxidized. The oxidized molecule is becoming more positive because it has lost the negatively charged electron. Hydrogen atoms are very important in metabolic reactions because they are the source of electrons, and we can think of them as electron carriers. When split, hydrogen atoms yield a positively charged proton and a negatively charged electron, which can then be transferred to other molecules. In this figure, molecule A has an electron that is transferring to molecule B. After the redox reaction takes place and the electron is transferred, molecule A is oxidized because it has lost the electron, and molecule B is reduced because it has gained an electron. Whenever a molecule loses an electron, another molecule has to gain that electron. To help you remember which molecules are oxidized or reduced, remember Leo the lion says GR. Leo stands for lose electrons oxidized. GR stands for gain electrons reduced. Chemical energy stored in the bonds of the glucose molecule are used to make ATP. In this overall equation of cellular respiration, we see that glucose and oxygen are used by cells to yield the products carbon dioxide, water, and energy in the form of ATP. We can see there are several hydrogen atoms that are part of the glucose molecule, but there are none in the corresponding molecules of carbon dioxide. If we think of the hydrogens as electron carriers, we can say that glucose is being oxidized to carbon dioxide because it has lost its electrons. Conversely, oxygen is being reduced because it has gained electrons from incorporation of the hydrogen. We see the gain of hydrogen atoms in going from oxygen to the water molecules. We can also classify metabolic reactions based on whether energy is consumed or released during a reaction. Like redox reactions are coupled, so are endergonic and exergonic reactions. Endergonic reactions require the input of energy, and exergonic reactions release energy that can later be used in an endergonic reaction. Doesn't that make sense? In this example, we see the glycogen molecule at the top is a string of glucose molecules joined by chemical bonds. Recall that chemical bonds store energy, so there is more energy stored in the glycogen at the top than in the individual glucose molecules at the bottom of this figure. In the exergonic reaction, the bonds in the glycogen molecule are broken and energy is released. The energy that is released can be used in an endergonic reaction to form bonds between the glucose molecules. Because the glucose molecules are moving from a state of disorder back to a state of more order. So from a state of high entropy to low entropy, energy is required. ATP stores energy in the form of chemical bonds between the phosphate groups. When the outermost or that third phosphate group is released, energy is released that can be used to do work in an endergonic reaction. And then ATP can be regenerated by gaining a phosphate molecule. The chemical energy stored in the ATP molecule is used to run metabolic reactions. Metabolism is the sum of all reactions of an organism, and these reactions are either synthesis or anabolic reactions, or degradation or catabolic reactions. Enzymes help metabolic reactions proceed faster and with less energy. Enzymes are a type of protein, and some are located in the cell membrane. They may form metabolic pathways that specifically convert substrates to products. In this figure, there are 3 enzymes embedded in the cell membrane. Enzyme A specifically binds the substrates and converts them to the first product. The first product then becomes the substrate of enzyme B, which converts that substrate to the second product. Product 2 then becomes the substrate for enzyme C, and enzyme C converts it to the final product. Products 1 and 2 are called intermediate products because they are not the finished final product of the metabolic pathway. In the cell, intermediate products are necessary because they can be rerouted to other metabolic pathways to create different final products. When enzymes bind substrates, they lower the activation energy of the reaction. Enzymes provide a meeting place for the substrates, so they are more likely to be converted to products than if they were just randomly floating around the cell. The top of this figure shows a man pushing the lactose substrate up a hill. Once the lactose gets to the top of the hill, it's going to be converted to the products, glucose and galactose. But it's going to be converted to those products, but only once it reaches that top of that hill where the activation energy has reached its peak and the substrates can now be produced. The activation energy is the amount of energy it takes the substrates to get to the top of the hill where they can be converted to products. The net energy is the amount of energy released from breaking the bond in the lactose molecule. Comparing the top figure to the bottom figure, it takes much more activation energy to get substrates to the top of the hill without an enzyme than it does when that molecule has an enzyme to bind to. We can subtract activation energy from net energy to determine the amount of energy remaining to do work. So, doesn't it make sense that if we use less activation energy by using an enzyme, there is more energy conserved to do work with in the cell? Enzymes are a type of protein. Recall that proteins must be folded properly in order to function properly. In the case of enzymes, protein folding forms pockets called active sites that are perfectly shaped to fit its specific substrates. In this figure, the enzyme is folded to form two active sites that perfectly fit the shape of the substrates. After the reaction occurs, the products are released from the enzyme and it is free to bind more substrates. It's important to note here that enzymes are not consumed in the reaction, but they can be reused until the cell degrades them. This is the end of part 1 of metabolism. Thank you for listening.